Okay, so today we are moving on from the stuff we talked about at the beginning of the class, like the first half, the Mendelian and transmission genetics, where we're talking about whole chromosomes and genes moving around and recombinations and things like that, to the part of the class that deals with molecular biology or molecular genetics. So we're going to talk about the structure of DNA, and we're going to talk about how DNA is replicated, more copies are made. So the biochemistry of what happens during S phase that we've talked about earlier in the semester, and also how what is encoded by the DNA molecule becomes proteins in the cell. So we're going to talk about how a single-stranded RNA copy of DNA is made in the process of transcription. Stop moving. And how this is translated from the language of nucleic acids to amino acids to make proteins. And so I thought I'd start with a quote from Watson and Crick's famous Nature paper from 1953, where they planted the flag, so to speak, with regard to solving the structure of DNA. If you read this paper, it is kind of amazing. It's like a 2-page paper, and it was a Nature paper for which they won the Nobel Prize. Things were different back then. The paper doesn't have much more than a sketch of DNA and a little description of what it is structure-wise there. The evidence for how they solved that came in later publications, and journals used to let you get away with that. They do not anymore. You have not only multiple page papers, but usually a lot of supplemental material with it anyway. So from the last part of the paper, this is the big deal. This is why solving the structure of DNA was so important. It says, "It has not escaped our notice that the specific pairing we have postulated, which means the A's pairing with the T's and the G's pairing with the C's, and as we'll see, the way that the two helices are intertwined with each other, the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material," meaning that if you have a DNA molecule, you can take these two strands apart and each one can, and it turns out will, be used as a template to make the other strand. And also it turned out later that the DNA strand is the template for making RNA strands. Now translating from nucleic acids of RNA to amino acids of proteins is a little bit more complicated and took a lot longer to solve, but once the structure was known, it seemed like the door was just busted wide open as far as figuring out how all these processes worked. All right, so what are we going to do here? If you want to use the genetic material, we know that's DNA, but it wasn't known at the time. So what are the properties or characteristics of a substance that will function as the genetic material? So first, pretty obvious, it must contain complex information. So written into the 3.02 billion bases of your DNA are the instructions for going from a single cell, the fertilized egg, which ends up being the zygote, to an entire being. Have to replicate it. So you start out with one cell, you end up with, like, I don't know how many trillion cells are in us, but a huge number of cells. So there are a lot of mitosis processes that have to take place, and before each one you have to have S phase. You have to take the DNA that's one copy and make 2 copies out of it. You can't make too many mistakes or things won't go well. You have to be able to, from the genotype, the DNA sequence, get to the phenotype. And what I just described earlier is still referred to as the central dogma of DNA. So DNA gets transcribed into RNA and gets translated into protein. Since then, there have been a number of corollaries coming to it. So as we'll see, there is the ability to have reverse transcription, so you can actually use RNA as a template and go back to DNA. And it turns out that RNA can actually be replicated or copied, so you can make another copy of RNA out of a first copy. And it must allow the evolution of the organism. So numbers 2 and 4, kind of like the scales of justice, right? You want to have a balance between new mutations on one hand and accurately copying the DNA on the other hand. If you allow no new mutations, so if the copying of the DNA is completely faithful, like there's no mistakes made at all, then there won't be any genetic diversity in a population. That is a bad thing, because when environmental conditions change, then if those conditions become non-conducive to growth for the population, the genotype of the population as it exists, if there's no individuals within that population that have a different genotype that can survive in the new conditions, then that population dies out. But, which did I say, this is mutation on this side, right? So you have to have enough mutation. But if it's tipped too much like this and you have too many mutations, then most mutations don't make any difference to the organism. They're neither they neither help nor hurt with survival in a certain environment. The second most popular class of mutations are mutations that cause a negative phenotype. So all the mutations you're looking at in the lab, they're curiosities that we use for doing genetic mapping, and they're very useful in the lab, but these would never be tolerated in a wild type population because paralyzed, rolling, the short and fat can't move very well. All these mutations would make the organism that is homozygous for them less fit to survive and reproduce than organisms that don't have them. So they would be, the wild type organism would be selected for over the organisms that have these mutations. Yes? Okay, so I've talked about some of the best examples are the ones that kind of balance out, right? And have I talked about sickle cell in here yet? No? Okay. So, sickle cell anemia, what's that? Who knows what that is? Nobody? Yeah? It makes your red blood cells instead of spherical or sort of disc-shaped into a sickle shape that will cause problems because they plug up the capillaries, especially at your joints. This is a bad thing. People who have sickle cell are not in good shape. Now, but it's incredibly prevalent in individuals that come from sub-Saharan Africa, and a lot of, used to be the Caribbean, but not so much anymore, and a lot of areas of the subcontinent of Asia, so India, Bangladesh, places like that. Why did this mutation that when it's homozygous causes so much problem get selected for? So see, it's negative, right? You would think it's negative. It turns out that if you are heterozygous for this, If you're a heterozygous, this is on a downward slant. If you're heterozygous for this, it turns out that you have a greater resistance to infection with the parasite that causes malaria. And the areas that I've just mentioned, sub-Saharan Africa, used to be the Caribbean, and I think still is to some extent, and around the coastal and warmer areas of India and Bangladesh, these are areas in which malaria is still present. So malaria used to be a problem in the United States, especially around New Orleans area, but we've kind of wiped it out. In these other areas, it's still prevalent enough that it's natural selection, and these mutations are selected for because the negative effect of being homozygous for this mutation is balanced out by and eclipsed, in fact, by the positive effect of being heterozygous for this mutation. All right, so there's another one that we'll talk about. Does anybody know anybody, or everybody has probably heard of cystic fibrosis, right? Does anybody know what that is? The medical condition for that? Cystic fibrosis is a mutation in a chloride channel. Remember back from introductory biology, the ion channels that allow ions to pass in and out of the cells. Usually they talk about the sodium-potassium pump and other channels that allow free ion flow. Well, this is a channel that allows chloride to flow, so it maintains the chloride balance, especially in the epithelial cells of your respiratory tract. If you are homozygous for a mutation in the cystic fibrosis transmembrane conductance regulator gene, CFTR, you make channels that don't regulate the amount of chloride ions in your cells correctly. Your chloride balance goes out of whack, your sodium balance goes out of whack, and what happens is these individuals get a buildup of mucus in their respiratory tract. Treatment has increased greatly. There's medicines that will, proteases that will digest out the mucus or phlegm, and there's also mechanical mechanisms by which these people who are affected get, you put them over somebody's lap and like whack the bejesus out of their back. That looks like you're torturing them, but what it does is the combination of the proteases and this manual disruption allow the mucus to break up and be coughed out. Because if you don't, these individuals get infections and die, not from the mutation itself, but because you get the mutation accumulating, right? So this is in a different population. The CFTR mutation is, I think, about 1 out of every 2,500 Caucasians who are from what used to be, who are from this, formerly their ancestors are from the Central European area. So does anybody know, besides the bubonic plague, what was a big bad killer of people in that area? Cholera, right? You get cholera from contaminated water, and it's you get diarrhea to the point where you can't maintain fluid balance and electrolytes, and you die from that. While being homozygous for cystic fibrosis transmembrane conductance regulator gene gives you cystic fibrosis, which at the time was deadly well before reproductive age. Being heterozygous means that it doesn't prevent the infection, but it helps you to survive if you get cholera. And so again, it's a balanced out issue that the positive selection that occurs when you're heterozygous, if most of your village gets cholera and dies and you don't, but 25% of your kids will get cystic fibrosis and die, that's trade-off evolution makes every day, right? but then now that those problems are gone away, you know, let's look at populations in the United States. You know, those certain populations that I just mentioned, for each are still carrying around a very high level of these mutations. And the negative selection is still there, but a number of mutant alleles is so high that they're just not going away. Even though the positive selection ended a long time ago. So nobody gets, well, malaria is very unusual in the United States for people who have not picked it up somewhere else. And cholera is almost, is very, very, very low occurrence in the United States. So the selection isn't gone. So there's two examples that are like a balancing out. So along the coasts of one more example, like along the coasts of the country where there's a lot of flooding from the ocean that occurs naturally. If you are a plant that obtains the ability to grow in conditions of higher salt, you will be favored there as well, right? Things like that. And it's very small differences that make very small advantages when considered across an entire population can push things along quite rapidly. All right, so I'm going to talk, and let me take this opportunity to tell you that if you want to know more about this topic, then sign up for Evolutionary Biology with none other than yours truly this spring. It's going to be an early class, so the time we could get it in was like 8 on Mondays and Wednesdays. But you get it done with, and then you have the rest of your day to do things. So consider signing up. All right, so I'm going to talk about a number of experiments. That established the fact that DNA was in fact the genetic material. And it seems obvious now that, well, of course it is, how could it not have been? But in the past, things were kind of murky, and as we'll see, there are reasons for thinking that proteins and not DNA were actually the genetic material. So let's start off with the transforming principle experiment. So what do I expect you to know for these? I'm not going to ask you for the exact date of each experiment, but you should know which experiments occurred in which order, and once we talk through them, it's somewhat easy to remember that. What I do want you to know is the details of exactly how the experiment was done, and also what happened at the end and how the conclusions were drawn. So here, a physician named Fred Griffith was working with mice, and he was working with virulent and non-virulent strains of the pathogen Streptococcus pneumoniae. Who's taken microbiology? Somebody explain the difference, what's virulent and what's non-virulent? Right, yeah, one makes you sick and one doesn't. And in fact, in this case, the type 3S, as you can see from, if you get injected with that, you're going to get sick and you're going to die. The type 2R, you are not. The S refers to the colonies looking smooth if you grow them on a plate. The R refers to the colonies looking rough, so a more rough and less smooth appearance if you grow them on the plate. Also, microbiology people, does anybody remember what a capsule is? Can somebody tell me what a capsule is? It's what protects the, well, okay, a capsid, C-A-P-S-I-D, is the protein that goes around a virus. A capsule is a coating that protects bacteria. So generally made out of sugars or polysaccharides, but can also be made out of proteins. It is a thick layer that surrounds the surface of the bacterium. And the reason why this usually makes bacteria more virulent is it insulates the bacterium's cell surface from our immune system, and so we can't get at the cell surface antigens and have an immune response against them. So Griffith was able to grow the virulent type 3S bacteria in a flask. When he injected the mouse, it dies, and then from the dead mouse, he was able to recover bacteria that could be grown up in a flask again and injected into another mouse, and it would kill them. Anybody remember Koch's postulates, or Koch's postulates, depending on, right? That's from microbiology, that's exactly what this is. Koch's postulates are what you use to identify and show the cause of an illness and prove it. So you have to be able to use the organism to cause an illness, show that the illness, in this case death, happens, and then you have to be able to recover the same kind of bacteria from that. And we got that here. Type 2R, non-virulent. These are the same species, but it's just missing a gene it's missing a gene that's necessary to make the coat or the capsule. You can grow them up, but when you inject them into a mouse, because the immune system is able to recognize the bacteria, you'll mount an immune response, and the mouse may originally get sick, but then it will, once the immune system gets a hold of it, take care of the bacteria, kill them all off, and the mouse lives, and as a result, you don't get any of the bacteria left because the mouse cleared them all. The bacteria are all gotten rid of. But here's where the weird things happen. So Griffith showed that you were able to take these type 3S bacteria, expose them to heat for a little bit, but a low enough level of heat that they won't grow anymore, so you've killed them off. Inject them into a mouse. Inject them into a mouse. The mouse lives because the bacteria are dead and can't grow. You're just injecting dead cells, and no bacteria are recovered. In fact, this is the way a lot of vaccines work. They either use heat-killed versions of bacteria, or they use weakened versions of bacteria that are alive but don't have the capability of causing illness. So they're weakened in a way that makes sure that while they try to replicate a little bit, the immune system is going to get them, and as the immune system gets them, you develop memory B cells that will save the memory of that bacteria. So if you ever see the real thing, you can mount an immune response very quickly. All right, but the weird thing happened when Griffith took the cells that were not able to cause infection and the dead cells that, if they were alive, could kill the mouse, but now they can't because they've been heat killed. You mix them and grow them for a little bit, inject that into a mouse, and the mouse dies. And interestingly, live type 3S bacteria are recovered, even though the only live cells that were put into this organism were the type 2R. So you're mixing type 2R that's alive, type 3S that's dead, put them into the mouse, and somehow a transforming principle that he could not identify had allowed the type 2R strain to be transformed into the type 3S strain. And of course this was repeated over and over, so it was not a fluke. Everything was done okay. But what is this transforming principle? And interestingly, we're going to do, not this experiment, but we're going to do DNA transformation in a couple of weeks in the lab, where I'm going to give you some DNA and you're going to put it into bacterial cells so we can grow it up. This process is called transformation because of this experiment. So whenever you add DNA to cells and it stays in there and changes the cells, you're transforming the cells. Yep. Yes. So the question is this horizontal gene transfer? Yes, because, I mean, I don't know if it truly would count because these, the type 3S cell or type 3S cells are killed, so it's not like conjugation. It turns out that the type 2R cells are, and I'll show a slide about it in just a second, but the type 2R cells are actually picking up DNA from the killed type 3 and they're taking it for themselves. So I think that would count, but I don't want to go out on a limb. Yeah. Is what? This is natural transformation. So the transformation we're going to do in the lab is artificial because some cells are what we call competent for transformation. That just means they have the ability to pick up DNA and take it in. These cells have a natural competence. We're going to do transformation with E. coli cells in the lab, and they have to be chemically treated to weaken their cell walls, and then we're going to stick them in a hot water bath to heat shock them to allow them to be transformed. So E. coli does not possess a natural ability to be transformed, but we can treat them chemically and make that happen. All right, so this is what I was talking about earlier. What was known at the time, and why would we not suspect, like, hey, why isn't it just DNA? So we knew nuclei carry the genetic material in eukaryotic cells. Remember, prokaryotes don't have a nucleus. Deoxyribonucleic acid, or at the time known as deoxyribose nucleic acid, chemists knew that it consisted of a polynucleotide chain. They did not know about this structure, obviously. Analysis of nuclei from eukaryotic cells showed that they contain both protein and nucleic acid. And in fact, the ratio is about 50-50. So if you take chromosomes from a eukaryotic cell, they are composed of about half protein and half DNA, nucleic acid. In chapter 11, we'll see exactly what that is when we talk about the packing of DNA into chromosomes. All right, so which of these, if either one, was the transforming principle? And so at the time, the structure and composition of amino acids or proteins was known as well. Not the three-dimensional structure, but what a chain was composed of. So DNA, as we'll see, is composed of sugars and bases and phosphates. Proteins are composed of amino acids that are linked together with peptide bonds. It was also known that there were 4 bases that are commonly found in DNA, and there are 20 that are found in proteins. And so if you were trying to write a book that is able to show or able to have the instructions for making an E. coli bacterium, a mouse, an elephant, or a person out of one cell, would you have more success, do you feel, writing a book with an alphabet that contained 20 letters or an alphabet that contained 4 letters? 20, right? Because it's more complex. How the heck can you do that with 4 letters? It's amazing that it works. It does, but it wasn't obvious at the time. All right, so we jump from 1928 all the way up to 1944, so not all experiments shown, right? But this famous experiment by Avery, McLeod, and McCarty demonstrated that it was actually DNA and not RNA or a protein, the other common constituents of the nucleus, of the eukaryotic nucleus, that is the transforming principle. So they did the same kind of experiment that was done earlier by Griffith's group, except here they took the type 3S virulent bacteria, heat kill the cells, and then essentially blend them up so that mix them up so you have this bacterial filtrate. So you lyse open the cells and pass them through a filter, and this is important, a filter that is so small that no cells can get through it. So this isn't not only heat killed cells that can't grow, but this is the lysed up soup that comes when you break the cells open, and there's guaranteed to be no whole cells, live or dead, in that filtrate. They showed that this filtrate contains the transforming principle by doing the same experiment with the mice that Griffith's group had done, and then they repeated the experiment with a separate set of treatments. So they took the filtrate and split it into 3 different treatment groups. One was treated with the enzyme RNase, which will break up an RNA chain into the individual nucleotides. Protease, which will dissolve the bonds and break up the bonds that hold the amino acids together, so you go from being a polypeptide amino acid chain to just a bunch of amino acids, and DNase, which breaks up DNA into the individual subunits of the polymer. Then they added each of these to type 2R bacteria. With the RNase and protease treated flasks, they ended up getting type 3S bacteria that are live, being able to be isolated from that. And when these were injected into mice, they killed them, as was expected from the previous experiment. However, with the type 2R bacteria, when the DNase treatment was used, they only found type 2R bacteria, no type 3S, and also these, when injected into a mouse, had the same effect as untreated control 2R cells. They did not cause any illness, and the mice lived, and no type 3S was ever seen there. Each of these treatments only gets rid of one of the 3 possible transforming principle candidates. The other 2 are still there. So this one that failed to transform the cells had RNA and had proteins, but did not have DNA. And so by doing this, they demonstrated that it was DNA that was the transforming principle. But because of what I was talking about earlier, there were still a lot of doubters out there. I don't know why. This is a pretty convincing experiment, but there were still people that were thinking, well, I don't know, they must have done something wrong, or who knows what. I think that this should have been it. All right, so here's, you're asking about natural transformation. So I picked up this figure from the chapter that we don't cover, the bacterial genetics chapter, but you should have talked about transformation, or will, in microbiology if you take that in the future. If a cell is competent to be transformed, it has the ability to pick up DNA from the environment, from other cells. In this case, the heat-killed cells had seeped out a little bit of their DNA. You know, the cell wall was weakened a little bit by the heat-killed treatment. DNA is double-stranded, but in the transformation process, only one strand goes in. And then, because it has homology to part of the genome because it's from the same species, a recombination reaction occurs. And you're like, and so you may be surprised to hear that recombination actually happens in E. coli cells. And that's where recombination in eukaryotes is derived, from where it's derived. Right? It's like, why would you need recombination in a cell that doesn't have more than one copy of its genome? This is one of the reasons. To be able to take DNA from somewhere else and do this horizontal gene transfer. So transformation in this way, or conjugation where there's a pilus or a little tube that actually gets made and extended between two bacteria, and one bacterium just directly sends some DNA down to the other one. Both accomplish the same thing. So here, because this match is part of the genome, it goes in, and then you have the original strand here in green and the new strand there in orange. After replication occurs, you have one copy that has double-stranded of the new version. And that's what happens. So E. coli has the ability to do recombination just fine, but it does not have the ability to do this without us beating up on the cell wall by having a harsh chemical treatment and then heat shocking it. Make sense? So now somebody should be asking, if E. coli doesn't do this naturally, I think E. coli can do conjugation, so that's fine. But if E. coli doesn't do this naturally, you know, what's another reason why it would have the recombination capability? The answer is because, as we'll see in chapter 12, the DNA replication chapter, E. coli, when they're growing under optimal conditions, can divide about every 20 minutes. So one cell division every 20 minutes. It's incredible. It takes longer than 20 minutes for the E. coli cell to copy its DNA. So in order for the E. coli cell to be able to replicate at that super fast rate, it starts copying the DNA again before the first copying is finished. So oftentimes, I mean, it's not homozygous or heterozygous because there's only one chromosome, but in some E. coli cells, certain parts of the genome, there can be 3 different copies present at the same time, because you have the first copying almost done, the second copying is about half done, and the third copying has actually started. And if any bad stuff happens, like a chromosome break or ultraviolet light damage has happened, the E. coli cell can actually use one of these extra copies to fix one of the other ones with homologous recombination. All right, so this one is the final straw, the final experiment. So we went from 1928 to 1944. I mean, a lot of time passed, right? 1952. This is only 1 year before Watson and Crick published their structure of the DNA molecule. So it's called the Waring blender experiment because that's the brand that they used, and so that's what I have at my house, and I'm the only one that cares. But I, you know, have to have it for that reason. Got to nerd out a little bit. All right, so in order to understand the Waring blender experiment, you have to understand the replication of what's called a T2 bacteriophage. So those of you who have taken microbiology, what is a bacteriophage? What? It's evil. Oh. What do they do? They inject themselves into the bacteria, they replicate, and then they destroy the bacterium from the inside. Does the whole T2 bacteriophage goes in? Just the DNA. Hence the whole experiment. So bacteriophage So bacteriophage, what does phage mean? Eating, right? So these are bacteria eaters, because if you infect a culture of bacteria with bacteriophage, it goes from being a cloudy culture, indicating there are a lot of cells there, to a clear culture. I think they show you one of these, if they still do that, in Principles of Biology 1 lab. The last time I did it, they showed you, here's a tube full of cells and you can shake it up and it's cloudy. Here's another one that's been infected with bacteriophage. It's clear because the cells have all been lysed or broken open. So bacteriophage is like a virus for prokaryotic cells. And that's exactly what it is. Around the end of World War II, there were a lot of physicists who formerly, and I don't know how this happened, but it's the truth, a number of physicists who were formerly working on the Manhattan Project, and once that was finished and the ability to make atomic weapons was established, they didn't need nearly as many scientists working on that, so these guys were looking around for something else to do, and a lot of them actually got into the bacteriophage game. So bacteriophage is a model organism as well, because it's very simple. It contains DNA inside of it. It infects bacteria. The phage goes on to the outside of the cell, and the bacteria are injected in sort of like a syringe. Then on the cell surface, these protein ghosts remain attached. They're called ghosts because they don't have the DNA anymore. Their soul, so to speak, has gone into the cell. Then the bacteriophage, as was stated absolutely correctly, hijack the cell's machinery. Just like viruses can't replicate on their own and therefore are not considered alive, neither are bacteriophage. But they use the bacterial cell's own replicative machinery. At the same time, you notice this is the bacterial chromosome. It's circular. It chops it up. So it's like it shreds the bacterial chromosome, making the bacterium sort of like a zombie that is just there to do the bacteriophage's business. And it makes the DNA replicates, and it also makes more protein coats. Bacteriophage are self-assembling. So if you take the pieces of protein coat that make the capsid and the bacteriophage DNA and throw them in a test tube, they will spontaneously form new bacteriophage, and that's exactly what they do in the cell. And then when the bacteriophage are done, their coup de grace, so to speak, is they force the bacterial cell to lyse open, and out comes about 100 new bacteriophage ready to infect another cell. And that should be a 4, not a 1. And that's why you get this clearing of bacteria. You go from a cloudy culture to a clear culture because all the cells are being lysed, and when they're all gone, that's it, and you have a whole bunch of bacteriophage here. But they can just hang around, right? I used bacteriophage when I was in graduate school, and you don't even have to freeze them. You keep them at 4 degrees, which is just refrigerator temperature, and they're good for years and years and years. Sometimes some of them will die, but not a lot, and you can get them growing again, and it is perfectly fine. And so scientists were using the replication mechanisms of these as a model system. That's not what excuse me, that's not what Hershey and Chase were doing here, but what I'm telling you is that the T2 bacteriophage system was already fully established as a model system. So when you throw bacteriophage in with E. coli at a certain temperature, scientists knew exactly how long it would take for the bacteriophage to latch onto the outside of the cell, exactly how long it would take for the DNA to be put into the cell, exactly how long the process of making new bacteriophage would take, and when the cells would burst. It was, timed down to the minute. And so, just like the nucleic acids of the eukaryotic cell nuclei, phage capsid, all these parts here, are made out of protein, and the DNA is contained within, but weight percentages are almost exactly 50-50. Right? So what they did was they did 2 treatments of bacteriophage, so 2 completely separate experiments. And they used a technique called radio labeling. See, this is taking all your classes into account, right? Does anybody remember radioactive? Oh wow, look. Sorry. Does anybody remember radioactivity from Chemistry 1? Yay! Or I think it might be in Chem 2, but either 1100 or 1200. What is radioactivity? It is when a nucleus is unstable. So usually in a nucleus, you have the same number of protons as you do neutrons. And in some cases, it's possible to make nuclei that have too many neutrons compared to protons. Some of these, as we'll see in the replication chapter, are stable, meaning they can just exist as an atom that is too heavy because it's got 1 extra proton in it. But a lot of these are what's called radioactive. They're unstable. The nuclei at a certain rate will break down into other nuclei, releasing some kind of energy, either alpha particles, beta particles, gamma rays, what have you. And these radioactive decay events releasing these particles or waves allow that radioactivity to be measured and monitored, so you can see if there's decaying. So they decay at a certain rate, but it lasts long enough to do the experiment. All right. As we'll see, DNA contains phosphorus because every one of these sugars is joined by a phosphate. So the purple atoms here on the DNA model are all phosphorus. So DNA contains phosphorus, but the backbone and the bases contain no sulfur. 2 amino acids contain sulfur, but no amino acid side chains, nor does the backbone of protein contain phosphorus. And so scientists took advantage of this by using 32P and 35S. These are radioactive isotopes. And I'm trying to, I can't remember, so they all have too many neutrons in them, but I'm just remembering that this is not a science classroom, so there's no periodic table. Nobody has one, do they? Well, I can bring that up next time. In any case, P32 and S35 are radioactive nuclei. They decay at a certain rate. So about half of the P32 will decay into other size nuclei in about 14 days. So that's the half-life. The half-life of S35 is about 60 days, so you can easily do experiments here. So these are 2 separate experiments. On the left, we're labeling proteins with S35. So you take the T2 bacteriophage, you take the E. coli cells, and you grow them for multiple generations in the presence of this radio-labeled isotope of sulfur. So the sulfur becomes entwined in all of the proteins of the E. coli cell and all of the proteins of the bacteriophage, which means the capsid around here. the capsid and the midpiece and the little tail fibers here. Everything except for the DNA. Likewise, in a separate experiment, the E. coli cells were used to propagate T2 bacteriophage for multiple generations in media that contained phosphorus 32. And that will radio-label the DNA. So the DNA of the E. coli cells and the DNA of the bacteriophage are all radio-labeled, or hot, as we say. Then they took the bacteriophage only, got rid of all the radio-labeled E. coli, and took the bacteriophage in both of these cases and exposed new unlabeled E. coli cells to the radioactive bacteriophage. And they waited just long enough, because they had it all timed out, until the DNA from the middle of the bacteriophage was injected into the E. coli cells. So the DNA was put into the E. coli cells, but the ghosts, the empty shells, were still on the hook to the outside of the cells. And then they put them into the Waring blender that is the namesake of the experiment, and blended them at a high enough speed that you got a shearing effect. So this shearing effect was able to knock all the protein ghosts off the outside of the cells, but not enough to homogenize the cells and break the E. coli cells open. So they did that for both of these, and then I don't know why they don't get a real centrifuge. This is a dangerous old style centrifuge where the rotor is just out in the open. Yes, I've used one of these. But they spin them down so that the cells will pellet to the bottom of the tube, but all the protein ghosts stay in the top of the tube. We good? All right. And here, there is no P32 or S35 in the media, so the only radioactivity coming in here is from the bacteriophage themselves. All right, and then they looked at what happened. In the case of the protein S35 labeling, there was no radioactivity in the pellet that had the E. coli cells. And when the E. coli cells were taken and allowed to grow up, they did produce bacteriophage, showing the genetic material had been added to the cells, but neither the E. coli cells nor the bacteriophage were radio-labeled. But what we call the supernatant, the part of the centrifugation reaction here that was not the pellet, the rest of the fluid, the liquid there, that remained radioactive because that's where the protein ghosts were. So this showed that the protein does not enter the E. coli cell. I mean, scientists had done electron microscopy to show that that was the case, but this shows definitively that the protein doesn't enter the protein doesn't enter the E. coli cells. In the P32 labeled, on the other hand, the E. coli cells were radioactive because they were full of the radioactive DNA from the bacteriophage. But the supernatant here that had all the protein ghosts in was not radioactive because the protein was not. These bacterial cells also produced new bacteriophage, and the bacteriophage were radioactive because they had the original phage DNA was incorporated into the new bacteriophage as well. So this proved it, and this is one of the, if you pick like the top 10 biology experiments of all time, this is going to be on that list because it was a very elegant experiment. You labeled the protein one way, you labeled the DNA the other way, and it showed that the DNA goes into the cells, but the protein does not go into the cells, and therefore the DNA has to be the genetic material because it's the only thing that goes into the cells. The protein doesn't. Yes? Why are they only using DNA and not RNA? I'll show an RNA example coming up. because eukaryotic nuclei have DNA and protein in, but they also have RNA in because the RNA is being transcribed from the DNA. Bacteriophage do not have any RNA associated with them at all. They have only the DNA genome and the protein. The only time there's ever RNA is, let me back it up here. The only time there's ever RNA is in these stages here when the phage DNA genes are transcribed into RNA by the E. coli's own polymerases and then translated into proteins to make the capsid pieces. Once they're assembled here and come out of the cell, there's no RNA in there. The red that I'm showing there is only DNA. And the purple shell or capsid is only proteins. So they're not able to make RNA by themselves, and they contain none. They use the cell to make RNA, then the RNA is translated into proteins that help to replicate the phage DNA and also make the pieces, parts, so to speak, of the capsid that assembles to make the final thing. So this is cool too because that was it. It's a, where's the picture of it, right? It's very simple. Everything shown in purple on this slide is protein. The red part in the middle is the DNA. That's it. And that's why it was a good model system, because it was the ultimate in simplicity. There's literally nothing there. I don't know how much DNA T2 bacteriophage has, but Lambda, a phage I've actually worked with, its entire genome is, I believe, about 55 or 58,000 bases. That's it. That's incredibly tiny. And so you have a very small amount of DNA that, although bacteriophage are not too complicated, still amazingly has the ability to hijack the E. coli cell, cause more of itself to be made, and then when it doesn't need the E. coli cell anymore, it just busts them open and bye-bye, you know, I'm on the way, right? So they are not really read like this, but this is an electron micrograph showing the end of an E. coli or bacilli, so this would be like the end of an E. coli cell, and all these are like the bacteriophage. So these are around the outside. All these are like the, if you were looking at this from the top down, just showing the capsid there. And somebody has taken this and artificially colored the background that yellowy brown color and artificially colored these that red color. So electron micrographs are not, they're black and white. Any questions about how this works? I mean, this was a really, what we in science call a really elegant experiment. So it showed, it was a great way to show exactly what was going on there. All right, so let me begin to talk about these two characters for a couple of minutes, and then we'll come back to it next time, and I'll let you go. So these are Watson and Crick. Here's James Watson. Here's Francis Crick. And there is their model of the DNA molecule. So these guys performed exactly no experiments to get the Nobel Prize. They were smart enough to take usable information that was available, and also, unfortunately, information that was stolen, and come up with the structure of DNA. So here's what they had to go in. Chemists had shown that DNA is composed of ribose sugars, so a 5-carbon sugar with an oxygen in the ring, which we'll show next time. Nitrogenous bases, so either 1 or 2 ring structures that had nitrogen and some other substituents coming off the rings. See here, organic chemistry. This is all the fun things all together. A scientist named Erwin Chargaff took DNA from a whole bunch of different species and was able to chemically analyze it to find out the percentages of each of the 4 bases present. And he found out that in different species, the percentage of the different bases varied, sometimes tremendously. But importantly, that the amount of A was always equal, close to being almost perfectly equal to the amount of T, and the amount of G was almost perfectly equal to the amount of C, like the molar amounts. Because A and G are 2 ring, and T and C are 1 ring, so it's molar, not the straight up weight. And here's where the bad news came in. Scientists were, and we're not going to be able to talk about it today, but I'll show you later, using a technique called X-ray diffraction, where they ran a beam of X-rays through a crystal. And just like when you run white light through a prism, it separates it out into all the different colors of the rainbow. When you run X-rays or electrons through a crystal of DNA or protein, it gives a diffraction pattern that can be used to interpret things. So the X-ray diffraction picture that they saw was from a scientist named Maurice Wilkins, and more specifically from a woman working in his lab named Rosalind Franklin, who was the only one, as we'll see, to be able to get a crystal good enough to give a diffraction picture good enough to make this judgment. But she was a woman in science, and they weren't treated too well then. And Wilkins, like, hey guys, look at this that this person in my lab is doing, like without telling her. So he brought them in and showed these 2 guys her lab notebook, and they took the information that was in it, and that was what allowed them, with all this other stuff, this was the key piece without which nobody would have been able to figure it out. They figured out the DNA is in this double helical shape. And then they used not a modeling kit like you have in chemistry class. Actually, once they knew what the shape of the DNA molecule was, and again, if they hadn't stolen it from Rosalind Franklin, they wouldn't have known at all. They cut out the bases, and you can't see here, but the bases are actually pieces of cardboard. So they took pieces of cardboard that modeled the bases and flipped them in all different orientations, like a jigsaw puzzle, because they knew how the chemistry worked, they knew how the molecule must be shaped, and they just kept flipping stuff around until they got something that worked. And then they published it. And Franklin and Wilkins published at the same time. It didn't turn out too well for her, unfortunately. And we'll talk about that a little bit more next time. So see you in lab today if you're on Tuesday. If not, I'll see you on Thursday.